Hi, sir.
Hi. Hi.
This is Aarti.
Yes, tell
So
me.
we have been informed that Dhruv and Badir are facing some technical issues, so they'll be joining in 10-15 minutes.
Okay, that's
So
perfect.
are you going to start?
Yes, I am.
Okay.
Okay.
Okay. So, okay, sir, we'll start now, and we will first tell the instructions to all the participants. So, Dr. Keerti will be telling you all the instructions.
Super.
Hello, good afternoon all. So welcome back for our tutorial session. And before proceeding ahead, there are some general instructions for the participants to follow. Like you have, for any doubts or questions, please raise your hands, or you can also put your questions in chat box, or can unmute yourself and ask questions directly. Hopefully you all signed up for Google Classroom. So yeah, let's get started and hope you enjoy this session. Okay, yeah.
Thank you. I think that was Kirti, yes. Yeah. So just to recap a little bit, in the earlier session, in the morning session, we spoke about a couple of things. Just going to remind ourselves. Basic image generation, particularly digital images. And we have been focusing on optical microscopy, obviously. Yeah? There are many issues which I do not touch upon because of their complexity with regard to electron microscopy, super resolution. There are many more issues, and today we won't be touching upon them. Suffice to say, there, if you want to read more, there is more literature on the topic. Right? The next important part that I touched upon was the so-called computational image analysis pipeline. And the reason I call it a pipeline is really because this idea that you go from your raw data, I'm sorry, not date, through a pre-processing step to the, if you're going to go by the segmentation route, through segmentation, which as we defined it is some form of deciding what is your signal and what is your background. Yeah. There are many fancier algorithms other than thresholding, and we haven't dealt with them today. But I think in some examples, Dhruv might cover something about it. Once you have that segmentation and you have essentially a list of points, yeah, point coordinates across time, you want to correlate them with the points in the subsequent time points. And for that reason, tracking is of vital importance. And we discussed a so-called search radius approach, which helps you link the points to their coordinate partners in time. And someone, I think, asked the question, will size decide it? Well, in a way, size decides it because there is a combination of the two problems. If green is T1 and red is T2, okay, maybe this was exaggerated. So X being the center of those points, then the primary factor that the search radius is designed to help you with is to find, and there are a couple of different approaches to this, to find all points that fall within that search radius R. And now, of course, if the object is very large, if the movement is dependent on the size, then it is likely to move less. So maybe there your R might matter as a function of size of object. What is much more important, and this is what I was trying to emphasize earlier, maybe it didn't come out clearly, is the extent to which it moves. So we'll call this maybe delta x, delta y, yeah, in a way the displacement. Because if it moves a lot, let us say this was not here but went out here somewhere, then your R will have to be wide enough to encompass that, and then you will start ending up in problems. At such a point, the designing of your acquisition time, meaning to say, if it moves this large a distance over the short period of time that you're observing, let us say your delta t, time interval between frames, was 1 minute, will need to be reduced to take into account the fact that effectively, if I were to think of this as a vectorial motion, then if the velocity v is say 10 microns per second, then by imaging at 100 milliseconds, I am going to be able to observe approximately 1 micron difference, which means I am essentially breaking my position into smaller bits, and I am able now to track with greater accuracy. So much more importantly than the size is the speed with which things move, right? Now for today, we have for the remainder of this hands-on session, we have a few things to go over. So I'm going to start with an introduction to Python. Sorry. Okay. Now, some of you did say that you already have used it before. And then we will continue with a worked example, which will be presented by Badir, of something called Kymography. And I think some of you already know what that is. But hopefully you will see a novel perspective, which is the quantification through tracking. Yeah? And the second worked example is going to be that of single particle tracking, which Dhruv will present, of beads in water which are a pseudo example of what happens when something undergoes a random walk. Because of course a cell will probably behave in, if you're looking at a chemotaxing cell, in a more directed manner, but in a way it's a little more challenging at a high density to look at granule motion and follow it, because if this works, this is your control case, then we have a high hope of this working too. Okay, so this is how we're going to structure for this afternoon. All right, now since all of you have signed up on Google Classroom, and I think you probably have access to it, I'd like you to click on the material called Introduction to Python, which should open a file which is referred to as Image2Num Python intro IPyNV. And when you click on it, you can use the connected app called Google Collaboratory. Okay, so I should show you what I mean. Sorry, give me a second. Okay, go back. Oops. Yeah, there we are. So Image2Numbers, classwork. So we talked about the lecture, and where are we? Introduction to Python. Okay, so this is what I mean. Click on this. Yes. Okay. And this brings up this window. And what you should be able to do is click on Google Collaboratory and see this. Okay, it takes a few seconds, so give it that much time. Yes. Now, if all of you have this screen, I'm sorry, if someone does not have this screen, please tell me. Yeah. Akshaya said Badhi is facing network issues. Okay, good. Sorry. Okay. So anyone who does not have the screen I'm showing, which is this code window which says image2num dash python intro.ipynb, please type in the window and let me know again. Oh, I should repeat it, Sajjan? Yes, of course, sure. So I went to classwork. So, okay, this is the course window. Yeah, you see the Google Classroom? Yes? Okay, now I go from that Google Classroom to classwork. Okay. There are a few things here. Okay. And in that, I click on Introduction to Python. It pops up this little material here. I can say View Material, or I can directly click on it. And then it gives me this option here, which I will not download. I will go to, oops, okay, there was some problem there. Okay, tab crash. Happens sometimes, don't worry. Nothing is broken. Okay, and Google Collaboratory. I clicked on Google Collaboratory, Google Colab. Okay. So if you're getting only as Sajjan is saying the message that only download, try clicking on this. The link is colab.research.google.com. Hi, Dhruv. And there you should be able to say open notebook or upload notebook. So if you download it, then you can upload it back again. So Sajjan, do you see the collaboratory screen, the one that I'm facing right now? Okay, give me a second, I'll be back.
Uh, Guys, if it's just a download option which is coming in the uppermost panel of the window, there is open with, and when you click there, there is connect, and you can type in collaboratory, and it will open in collaboratory for all those who are not, who are just getting the download option. click on the connect more apps. When that black color window with the download option opens up, click on open with in the topmost panel, and there is connect more apps, and just type in collaboratory in that, and then install collaboratory, and this window, whatever Dr. Chaitanya's window is showing up, that will show up. Just type in an yes if if we all are on the same page.
I'm not sure there is an installation required, but yes, I agree. You have to sort of connect the app because otherwise there is no point. Yeah. So I think maybe everyone's options are a bit different depending on what you already have as potential alternatives. Yeah. So if this is, I think, what Sarubi suggested, I agree. Okay. I think collaboratory. Okay, there. So you see, I think this is exactly what this person was saying. It's in my case already installed. In your case, it may not be. So you need to install it. So that those 3 dots should allow you to install. And if it is already installed, then just simply opening it. And again, I'm not going to uninstall it, obviously. I'm just going to click it. Okay. Okay. Anubhav said he could download and then upload it into Colab. So that's fine too. All right. So is anyone still having issues with this? Okay. I take that for a no. If there is a problem, please type in. Yeah, I have no problems. We just get this to work first for you. Do I take it that it works now? Okay. All right. So stop me if there's a problem, but I'm not going to start. What you're looking at here is a basic introduction to Python using this cloud-based computing platform called Google Colab. As it suggests, it's a collaboratory. It's exactly for these kind of purposes. And the editing of the code that you do is your own version. Basically, we've given you the code already. I think this is what we were hoping also as an aim, that this is your start point, your skeleton, if you like. Now, in this, I'm going to demonstrate a few aspects of Python programming so that you at least know what the more complex data structures that Dhruv and Badir have in mind to introduce to you. Okay. So in this particular sense, we're not going to deal with structural databases. Sorry. Okay. So you could use these simple skills to calculate characteristic protein sizes and so on and so forth, but not for today. So what we're going to do is set a variable a and b, and all of you who have done some programming at least know that we use variables because we want to generalize. You see, once you store a number somewhere, then you can do something with it. But if you're always giving inputs, and this is why I always tell everyone who works in my lab, create variables. Don't just simply put numbers in the parameters or in the functions, because otherwise you can't manipulate them later. This is somehow a very basic principle of good programming. And this thing, so if I use the hash, yeah, sorry, so it's going somewhere else. If I use the symbol hash, I can add text, which is this is a comment, i.e. not to be interpreted as code. And I click on this play button here, it runs it, yeah, but nothing changes in the output, is what you should see. So you see, it has taken 2 variables, a is equal to 4, b is equal to 10, and print is a command which I can invoke to print out the product A cross B or A asterisk B. Yeah. So I can also say something like the product is, and hopefully if this is, if I haven't made any syntax mistakes, yeah, okay. So you see that I can use a fantastic computer like a typewriter or a word processor. I can do simple arithmetic. I can calculate some simple numbers. I mean, not very useful. Okay. I can also make it say things, right? I can ask it to say, good morning, welcome to biology by numbers. And I can also use single or double quotes. And I can say the thing that C programmers were first introduced to, which is hello world. And there it shows up, right? So this is now using the print command for something that is here, which is usually the terminal or console. Now, if you were running it locally, and for those of you who are more experienced with Python, you know this, that you usually try to separate your console from your code. You write a code in some text editor and you put your or IDE if you are very fond of these fancy things. And your terminal is where your output is. Essentially, this is the code is your commands, and the terminal, these little blocks that you see as plain text, are your output. So of course, why write computer programs for biological problems? You're here in this course, so I assume you don't need to know this. You don't need this to be explained to you, but it is simplification of labor-intensive, time-consuming tasks. Those that could be error-prone and subjective, to make them more automated, universal, and confidence on the analysis that you're doing. It also helps with data collection, analysis, and representation. So basically, nowadays, I don't know if those of you who are familiar with a little bit of programming already know this, that journals at the time of publication, and most of you I think are PhD students, require you that you submit actually source code, especially for data analysis. In fact, it's got more exciting because many of the figures that are shown in very complex figures with statistical analyses and so on, they can be dynamically generated using this kind of an environment like Python-like environment. And a user, a reader, can then put in cutoff parameters so that the data is more dynamic. And this is a really cool development because of the movement to online publications. So at the end of the day, even as a wet lab biologist who, you don't care about programming and bioinformatics, you do need to know some basics, okay? Because so much is now turning into this kind of computational approach that it's like being able to cycle, okay, for a scientist. Okay. All right, so that's why you need at least some basic understanding of how to do this. And that's Python, in my view, is simple, easy, interpretable, portable, universal in some senses in terms of how many places implement it. Now Google is implementing it as a public service, as a collaboratory. So I think this is really useful. So what you can do with your code is, of course, automate the plotting or regression analysis, perform statistical analysis, or simulate your data or hypothesis. And in our case, we'll add one more item, which is analyze images. Yeah, analyze images and process them. Okay. So certainly, okay. All right. So I don't know if you noticed, but I can, in this kind of Python environment, put in code as well as text. And the way I'm doing it is that I am adding using, say, insert a text cell or a code cell. Sorry, just one second. All right, so I can add something here, which is just plain text. Yes, and this has nothing to do with any code. So okay. and this stays as text. This, as you notice, has nothing to do with print command, setting variables, or anything. I've skipped some of the other things. The idea is that you can use it as a massive calculator, link strings, do input-output operations, but we're going to go to more interesting things, so I just simply added this business. The kind of variables that you can use are numbers, strings, tuples, lists, and dictionaries. These are standard Python structures, and again, you'll see examples of these. Today in one session, we're not going to learn to become, I hope, I hope it helps you get an understanding, but I'm not sure whether all this will make sense to you. So please go ahead and read more. There's a lot of documentation for Python. So Python itself is probably, in my view, one of the best documented scientific programming languages. And it comes with something called NumPy, and this is something that you should make most use of. Okay, so you can divide the numbers. I mean, this is kind of going on and on about arithmetic. You can concatenate words and numbers, so you can sort of print only a part of a word. You see that in the square brackets, I think maybe it's not always visible what I'm doing. Yeah, okay, maybe this is easier. In the square brackets are the indices of the word, the term hello world, which was the variable, string variable that was set to A. So if you look at this, so let's say it's how many elements are there inside the inverted commas: 1, 2, 3, 4, 5, 6, 7, and 8, 9, 10, 11, 12, 13. So it turns out Python is base 0. So if we say this, it prints everything. You understand? If I say print 3 onwards, it'll take 0, 1, 2, and 3. So low world it will be, I hope. Yes, low world. Okay. So you understand what I'm doing here, right? I am essentially breaking the components of the word which was stored dynamically in this variable A into what are then the elements of the string. And using this sort of square bracket notation, the second colon at the end, at the end, indicates 7 onwards to the very end. I can also do it the other way around. I can do something like first three letters. Let us see if this works. Okay, hell. Okay, 1, 2, 3. The first 3. The first 5 will give me hello. Okay, got it? Okay, so I'm going to move on. So I can manipulate numbers, I can manipulate text, and I can create more complex data in the form of a list. List is a data structure that combines numbers in this form. Square brackets are the delimiters for it. I can put a bunch of numbers separated by commas and say print x, type x. Print x will print that array with the list, I'm sorry. Type x will tell me what is the nature of the variable x. What is its type? It's a list. It belongs to the class list, hence the answer that comes out here. Right? You understand that, I hope. If you have problems here, please, how is Python different from C or C++ except in the coding method? Oh, Snigdha, I'm not going to get into that today. Just a very simple, short answer. Python is an interpreted language, and C is a compiled language. You can write your the syntax of C and Python in some respects may be similar. Aspects of Python are even object-oriented. C and C++ are themselves different, and you can feel free to go back and read programming textbooks. But for the moment, my aim for you is to understand logic, algorithms, and data structures, and not to understand the difference between the different languages. Uske liye alag class lena padega. Okay, all right. I'm sorry, I can't help you any further than that. Okay, so not for today. All right, so then length of x, I can access it using n1 as a variable. I can store the length basically, and that's how I get some first element of list, last element of list. So please play around with this code because it's in your hands. This is exactly why I shared it with you. All right, and I can also give ranges, which are some values which is 1 to 11 with one interval. And I can create, so let us say I want to create a list of numbers between 1 and 5, yes, with a step of 1. Okay, but what you hopefully notice is that it took 1 as a start value, but it ended at 4. So as you can see, 5 is the non-inclusive upper limit. Yeah. So if you want 5 to be included, you need to add the increment itself. So I need to add 6. So if what I want to do is print out 1 to 5, then I need to add the upper limit as 6. That is the range function. That is how range works, right? And the data type of the term values, or let's call them V1, yeah, V1 is range. Okay, that's how Python recognizes it. And the length of the values is 5. Yeah, and I can also go iterate. So all of you have heard at least of a for loop, right? And this is essentially doing that. It's saying set an x variable x within the values v1, yes, in the range v1, and print those values x. Okay, and that's what we managed to do. Okay, we can also combine lists. By the way, Snigdha, none of this would be possible in C or C++. You would have to first define variables, create them, destroy them. There's a lot more overhead in programming in C, and it's much faster as a result. But if you have not learned programming before, then this is much faster because your learning curve is faster. I mean, literally, as you can see, this is, I mean, you're not doing anything, right? And it's 2 lines are giving you an iteration. Gunjan says, can I explain the loop part? Gunjan, all I did is I created a range of values from 1 to 5 using the command range 1, 6, 1. If I want to skip a number, I can change that increment to 2, let's say. So let's see what happens. Okay, so it creates 1, 5, 3 because I'm skipping every 2, right? Now what the for loop is doing is setting every element sequentially in the range. So I go back to maybe making it 1 to a number that is a part of that range. So I can also print the range. Okay, let's print the range because maybe it's not very clear from what I just said earlier. Okay, all right. So the range is 1 to 6, but to access the values within that range, I'm using this iteration system where I'm saying for x, which is an element in v1, sequentially go through it. This is sort of Python syntax, which is very, very simple and very powerful. You know, when you can do a lot with 1 or 2 sentences, that is what is called a powerful language. This is, in that sense, a high-level language, to answer Snigdha's question, I think. Whereas C and C++ are low-level languages. They need more syntax, more detail, more information, more commands to the computer to do things. Computers are stupid. You need to give the commands, but you need to be intelligent to code it, okay, and understand the logic. And that's really my only aim over here. I can also combine ranges and lists by squaring numbers and so on and so forth. So I can print range 6 by simply putting the variable 6. It goes from 0 to 6, and the values in that are in fact 0 to 6, and then x squared of that is taking the numbers. So I can also, so maybe I can do this, right? Okay, so it gave me an error, and that is wonderful. It says missing parentheses in call to print. Did you mean print x? So this is the first key of any programming language, which is debugging. You make mistakes, nothing is broken. The debugger actually tells you information, and there I have the correct answer. You see that this is something that in an experiment, it would take you 2-3 months to do. So there are some really nice things. I mean, of course, this is a 2-sentence code, 2-line code. If you have a 200 or 2000 or 20,000 line code, it's not that easy either. So usually when you're doing some scientific programming, that is how it is. Okay, good. So I modified the range. I squared the values. I can also use sets as containers. I'm sort of going to skip this because you have it in your hands and you can try it later. And I can also use if, else, and while statements. And these are sort of similar logic. There are some beautiful notes on scipy.org on how to use Python for mathematics, computing, and so on and so forth. And you run some kind of simple stochastic simulations. And for now, I'm going to stop and hand it over now to Badir. Okay. So wait a second. Before Badir starts, does anyone have any questions about the part I just covered? No, everything is clear. Okay, fair enough. I'm going to now, in that case, hand over charge to Badir, and I will stop sharing my screen. Okay. Okay. Great. So Badir, it's all yours.
So I can share the Python notebook on the classroom drive, right?
I would prefer it for the moment if you first demonstrate it, Badir, and
Yeah.
we'll discuss it later. We'll upload it at the end. Yeah.
Yeah, sure.
Yeah. Are you running it locally or on the collab?
Yeah, in the collab.
Very good. That's better.
You can see the screen, right?
Yeah.
Yeah. Hi everyone, I am Badir Hussain. I'm a project student at SciSets Lab, Dr. Chaitanya Aditya's lab. Is my sound clear? Am I audible?
You're audible.
Yes, we can hear you, Badir. Yeah.
So let me start with recapping the image basics, as you already know. So images are basically represented as a matrix, n by n matrix, with corresponding values to each pixels. So in an 8-bit image, each pixel can be represented by a value. That pixel value will be from 0 to 255. So any value between the 0 to 255 will be can be presented as a gray value for each pixels. Uh, uh, so in the first this tutorial, I'll be talking about how to generate a kymography plot, and to start with the kymography. So what is kymography? The kymography is a space time plot. The representing microscopy and microscopy series data as a kymography is useful to analyze the particle moment and to assess the displacement and velocity and other statistics from the particle moment. The kymography is basically a graph with two axes, a time axis and a position axis. So the chymography is used to analyze particle movement, which are predominantly in a single direction, say X direction. So we have seen one of the example in the lecture, the morning session. So we have seen the movement of vesicles inside a C. elegans axon. So I will show that movie again. So this is a C. elegans under a microscope. So here they have focused on the axon of the C. elegans, and we can see a movement of particles along the length of the axon. See these bright white particles moving along the axons are vesicles. So by using chymography method, we will try to quantify the movement and the trajectories of these particles. So each chymographs will be represented in a similar format as we see in this figure. So the x-axis will be the position or the distance of the particle, and the y-axis is the time axis. So the time will start from 0 on the top, and as time progresses, the chymograph will represent the intensity profile of a line of region in our image over time. So I will explain how the chymograph is generated in a while. This figure is the Kymograph analysis of the same video I have shown. So here the black lines and lines are actually the intensity values corresponding to the particles. And so we can see some of the particles as the time progresses, the position of the particle in the positional axis is changing. Some are moving towards the anterograde direction, and some are moving towards the retrograde direction. So here on the right side is a automated software used to analyze the chymographs and generate the chymograph from the microscopy data and analyze it automatically. So the software is called Amtrak, and it's automatically tracks the peaks of the particles and analyze the data to give out meaningful statistics. So this software MATLAB-based tool was developed in the CISL lab, our lab, by Anushri Shipalkar. And so I've put a link to the Amtrak software here. So for this task, I will be I'll be using Python to Python to generate the chymograph and track the trajectories of each particles, and then output meaningful statistics from that. So for the aim of this task would be create chymograph from the microscopy data. So we have the time series images of the axon particle transport, and we will use that to create a chymograph. And then after creating chymograph, we will detect the trajectories in that chymograph and extract the coordinates of the trajectories. So each particle trajectories will have a coordinates in the position axis, and so we'll use that data to analyze the particle trajectories. We will calculate the velocity and displacement of particles. So here we start with creating chymography, and the first first thing to do is to download the required data. So here I'm downloading the data. So I've downloaded a multi-stack image from the microscopy time series of the axon transport, and so I will use the multi-stack image to extract data points from that. So in the first step, so we have imported the required modules that we will use for this task. And so one of the important module we use is the CV2. It's a computer vision module. It is used to play around with images in Python. I use the CV2 or the OpenCV module to read or load the image data into Python, and this function will load the multi-stack image into our Python notebook. So these multi-stack image will contain all the frames of the data. So it will contain the frames from the first frame to the last frame inside. So the next step, I have created a list. As you know, the list will contain many elements inside it, separated by comma. So this image list, I've created an image list which will contain all the frames of the microscopy series data. And so as an example, I have visualized the first frame of the image series using these blocks of code. So if you want me to go through any code blocks again, you can type in the chat. So I will.
Yeah. I think Badir Snigdha, one of the participants, asked, can you please explain the modules part again? So I'm not sure what is meant by the modules, so maybe you can answer that.
Yeah, so so the modules are actually libraries of functions. It will have predefined defined functions inside. They are actually already coded functions. So we can access the library and use the functions inside the library or the module, and use that function, the ready-made function, to calculate anything or to so in this case, I'm using the OpenCV module for the first function. The CV2 is the OpenCV module, and I use that for loading the image data. And so these modules are already linked with the Python. And once I import the module, I can use any function inside that module. So I hope that answers. I will move to the next part. So here we can see the first frame of the microscopy series data. And so what we have to do is we have to select a region of interest in the axon. So since axon is a one-dimensional, can be reduced to a one-dimensional object, we can use a line of interest, and over that line of interest, we have to assess how the particles are moving, whether the particles are moving anterograde, retrogradely, or they are not moving at all. So for the next step, we will select a line of interest from the frame. So we know that images are basically a matrix with numbers inside, and so selecting a line of interest from an image will give a list like this. So each pixel in the line will have a gray value associated with it. So I will demonstrate how we can select the line of interest, and for after selecting the line of interest, we have to extract the pixel values or the gray values associated with the line.
Buddy, can you answer a question that Divya has asked, which is, can we import a video in any format or should it be specific? Dhruv, if you're there, you can also answer that question because I think you have more experience.
Yeah, so there's a CV2 function called video capture. So you can import like different types of formatted movies like MP4 and AVI into Python, but it's best to work with TIFF files. So if you have a MP4 or AVI, you can convert it to TIFF and then you can read it using this IM multi-TIFF function CV2. But you can read in MP4 and AVI files too. But essentially, these videos, they are made up of specific frames, right? So you'll have to be working with individual frames. So when you read AVI movies or MP4 movies, you'll be eventually storing each frame as a particular array or a particular matrix. Yeah.
Yeah, so we have to.
Just a second, Divya, does that answer your question? Okay, go ahead, Patish. Yeah.
So this block, in this block of code, we will select the line of interest, and then we will analyze the pixel values in that line of interest. So so in order to select a line of interest and its pixel values, we should first specify the coordinates of the start position and end position of the line. So I've used, so I have, here I have calculated the image coordinates manually using Image2. So I have the value for the column, I have value for the column numbers and the row number for the line I'm interested in. So the 249 is the value of the raw number I will use for the line of interest. And so I will change the column from 425 to 558. So here I have overlaid the line of interest I am going to use for the further analysis. Whenever you have got a different data, you can use the Image2 tool and look which portion or which line of interest you want to analyze and get and extract the coordinates of line using Image2 and use it here for further analysis. And this image here is the first frame as we saw before. And and I overlaid the line of interest that I'll be looking into for further analysis. So I will track the movement of particle along this line of interest, and so how their motion is along this line. So this graph plots the intensities along the selected line and intensity of pixels along the selected line. So the y-axis is the yeah. So here we have the pixel position on the x-axis on the line itself, and the intensity on the y-axis. So we can see that not all the pixels in the line are illuminated or have the same intensity, and the intensity vary across the pixels. So in the microscopy video we saw before, we know that the particles have a bright intensity and the background have a low intensity. We will use this information for further analysis. So this is for the this diagram is for the line of interest in our first frame. So we have to extract the pixel data for all the frames, and the same line in all the frames. And we will next we will iterate through all the frames in the time series stack.
Badir, I think there is a question from Sajjan, is
Yeah.
whether the above 8-bit image corresponds to the data from where you're selecting the line of interest. I am confused. And someone else also wrote, how does one select segmented line of interest using code?
Yeah, so if I can answer
Please, Dhruv, go
Satveer's
ahead.
question. So this Google Collaboratory framework is not compatible with many functions over OpenCV2. So any pop-ups that are available with CV2 that allow you to select ROI interactively, they won't work on Google Collaboratory. So that's why I selected them manually. So when like I'm coding in Python or when I'm coding in MATLAB, I can just show the image, and there's a function called ROI select. You just type it and then you can select a specific ROI, and it gets so you don't have to leave the coding framework. Yeah.
Yeah, regarding the question from Sajjan Rajput, so the 8-bit image I've shown here, it doesn't correspond to the actual microscopy image series data we are using. It's just a representative image of an 8-bit image. So I just use this for to give an example. So how would the data would look like once we process it? So I hope that answers. So in this section, we have extracted the pixel intensity data for first frame across this line of interest. So we will do that for every frame. And so when we can extract the pixel intensity value for every frame and store as a list. So this list will have many elements. So as many as elements as the number of time frames in our data, and the first each element will be another list which contain the pixel values along the line of interest. This block of code. So here I initialize a new list called K, and so the for loop here I will iterate through the image list. So as we know that image list contain all the frames for data in a as in a form of list. So I will iterate through all the frames, and then I will store the pixel intensity of the line of interest in the list we created here called K, and we can try to see how the K looks like. Yes, so so this is an element in the K. So this is one of the element in the list called K. So this element is actually another list which contain many numbers inside. So each these numbers will correspond to the gray values of the pixels in our line of interest, and the K will look like a list of lists. So I'll comment that out again. So we have actually extracted all the data we needed for visualizing the kymograph. So we have the intensity, pixel intensities for of our line of interest in all the time frames, and we just have to visualize the kymograph. So for the visualizing kymograph, I use another module called Seaborn. So this used to plot graphs and visualizing data particularly. So here the SNS.heatmap will visualize our kymograph. So we give the input data, which is K. K has all the pixel values for all the frames of the line of interest. And then we will plot the kymograph. So this is the kymograph we generated from the time series data. So the time axis goes from 0 to up to 250. The 250 is the number of frames in our data.
Could I request you to please increase the magnification of your screen?
Yeah.
Yeah. Thanks.
Is this okay?
Yeah, this is better. Yeah.
Yeah.
Okay, you just go to one line of the code. I just want to see it again because a bit higher would be better still. You increase plus. One or two more. Yeah, much better.
Yeah.
If any of you have problems, by the way, please feel free to type and we'll try to address it. Yeah. Go on, please, Badri. So.
Yeah, here is our time kymograph. So the y-axis is the time frame data. So it goes from the first frame to the last frame, and so we have plotted. So each these lines along the vertical axis will correspond to the line of interest corresponding to each frame here. So the x-axis is the position, the pixel position, and here we can see the intensity of pixels across all the frames, and we can clearly see that many of the pixels in each frame doesn't have any particles, or they are not there are no bright spots, and so there are many particle tracks we can identify from the kymograph. So this particular particle is not moving in the x direction or the position axis, and it's static in its movement. And this particular particle is start as a static particle and then move towards one direction. And so as the time frame goes from a lower value to higher value, the position of that particle is changing towards the left direction. So since we have the kymography here, we can actually qualitatively calculate the velocity of this particle and the velocity and displacement of this particle, or so for higher data, or if you are having too many tracks or trajectories inside the kymograph, we have to automate the process of analyzing the data. And so as a next step, I increase the contrast of the particle trajectories and the background. So what I did here is I specified a cutoff value of intensity towards the plotting function, and the plotting function will not show any intensity less than 120. And so I could make the contrast between the background and the particle trajectories even higher. So I will use so the next part of this task is to extract the coordinates of these trajectories automatically from this data. And so if when we get the coordinates of these trajectories, we can use the coordinates to plot the velocity or the maximum displacement that the particle has done inside the axon. So yeah, I'm not getting any notification regarding the chat. So if I want to explain anything, Dhruv, can you interrupt me in between if I want to explain anything to?
Yeah.
Okay. So for the next part of the task, we will detect and extract the coordinates of the trajectories that we have created from the kymograph we have created. So for tracking trajectories, we use another module called TrackPy. So we have to first install TrackPy to our Python notebook. And so since the TrackPy is not commonly used, so this will not be already linked to our collaboratory notebook. And we have to install it from other sources and then link to our notebook directly. So I will so here the TrackPy module is installing to our notebook. And so the first step in tracking the trajectories that we have created here is we have to we will again go back to the previous step. So as we know, the list we created, the K list we created here have the pixel values from every frame as its element, the pixel values of the lanes from every frame as its element. So for tracking the trajectories, the TrackPy will first look at the pixel values of each frame, the line pixel values of each frame separately. And so as a first step, it will look at the first frame, the intensity values from the first frame and look for the position of particle in that first frame. And it will do the same detection of particle position in every frame iteratively. And then we have to use another algorithm in the same module to link the particle movement. And so that's how we create the trajectories of the particle. This code does the particle location, locate the particles on the frame. So here I'm creating a new data frame or the with. So this data frame will have the location values of particles in. So the tp.locate function, we input the list here. The K0 is the first element of the list K. So the K0 have the pixel intensity of the line of interest of the first frame. So the tp.locate will go through its pixel values and locate the bright spots in these pixel values, the brightest spots in these pixel values. So the 7 we have given here is the width of the particle or the width of the object in pixel values. So this value we can manually look in our data how what's the width of the particle we are trying to track in the data and use that here. And so if we print the tp.locate data, we have so this is the first frame. So we have the the position of the particles that the algorithm located. So it has located 5 particles in the first frame, and the X position is given here, and the mass corresponds to the intensity or brightness of that particle. So we have its intensity here. So the end plot here, I've plotted the how the intensities of the detected particles are distributed over the first frame. So we have few particles which has a low intensity and which are less than 30 of this intensity unit. And so another argument to the TP dot location that's called mean mass can put a threshold value of intensity, and below which the algorithm won't detect the particle has any intensity below 30. So I've specified 30 here, and then did the TP dot locate function again.
Badir, there's a question in the chat from Amal Matthew. What if the particle was moving very fast and the intensity increased in the next 4 to 5 pixels, next frame 4 to 5 pixels after compared to, I don't understand the question actually. Amal, can you please be a bit more precise? I think I understand one part of your question, which is what if the particle is changing in intensity and moving fast? Yeah. So moving fast, it's very simple. If it's moving fast, you change your threshold. Yes. Would that be a fair answer, Badir, Dhruv?
Yeah. So till now I'm looking at so I'm getting a feedback from you, sir. Hello.
Sorry, what do you mean?
No, I'm getting a feedback to your mic.
Oh, I think I know why.
Hello.
I think like, Buddy, can you go to your Kymograph?
Yeah. Can you
So I
hear me
think
better now?
what Amal is asking is that if those tracks, if they like the track disappears for like 4 or 5 pixels and it re-emerges after a gap of, I guess, 4 or 5 seconds, will it consider it to a complete track or not? In
Yeah,
the
actually,
case of this Kymograph, so then it will
yeah.
not, it will break, it will give you 2 different trajectories. In
Yeah,
the case
actually,
of,
I'm
yeah,
coming to that part. So,
yeah,
as of now, we haven't looking at whole time series at the same time. We are looking at each time frame individually. So, we only care about each time frame individually, and we are looking at the how many bright spots are there in each time frame. So, I will come to the how the tracks are produced. I will come to the algorithm for that later in the part. And so we have some other arguments that we can change to consider the case the Amal said here. The TP dot locate, we use here, it just sports the bright features in all the frames separately. So here we use a threshold value to remove low intensity bright spot, and up to till here we did that only for the first frame. So the next is to do the same thing for all the frame. So the TP dot batch will take an input of the list of all frames. This will give all the elements of the list K. So the elements of the list K are the pixel intensity in each frame, and so that will iterate through the elements in K and store the data of located features or particles. And So I can show you how the F2 looks like. So here is the frame number. So on the 0th frame, so the algorithm has detected 4 particles, and so they have so the position, the x-axis position of the particles are also given here. So this is actually a very big file, and so the Python doesn't print that directly by this command, and so it only shows the first few entries and the last few entries. So there is a break here. So there are many elements in between these two numbers. So here they have shown the data for the last frame of the 250th frame. So there they have also detected a few particles, and their position along the x-axis is given here. So in this data, we have only a position of the x values. We have only the x values here. So I use this function to insert a value of y to this data frame. So we know that since we are considering a line as our region of interest, the particle will only move in one particular direction along the x-axis. So there won't be any motion. So we are not considering any motion along the y-axis. So the y will be 0 for all particles. So I insert a y is equal to 0 for all particles in the data frame. This changes to use the same data for the next coming steps. So as we saw here, we have detected the bright features or the particles in all the frames separately, but the algorithm hasn't connected these particles in each frame. So from just observing this data, we can see that this might be by connecting these lines, we will get the trajectory of the axial trajectory of the particle across different time frames. So the next step of the next step is to for the algorithm is to link the trajectories using the data we have created now. So the position of the particle data here. So as a next step, the algorithm will iterate through data from every frame, and it will link the particles, and so the linked particles will give us the coordinates for the trajectory of that particular particle, and I will go through the function to link the particle positions. So this tp.link function will link the particle position, and so we have to give the input data frame as F2. The F2, as we know, F2 is the data frame I've shown you here. F2 has the position of all the features in all time frames, but without the linking. So this tp.link will link the particles in across different frames, and the 4 here is the maximum displacement that we observe the particle in the time series data can take between a time frame. So I think by there was a earlier question regarding how we connect the tracks of particles across time frames. So one input we give is the maximum displacement. So say we are considering this particle on the F1 here, and so really they specify a number of pixels, the maximum number of pixels a particle can move within a time frame within a consecutive time frame. So if we specify a pixel length greater than the length between the second particle in the second F2, these two particles will be linked by the algorithm. But say if you are not specifying the maximum pixel displacement, if we are specifying a lesser maximum displacement, this particle won't be connected towards this particle by the algorithm. So we have to again manually look at how the maximum displacement of the particles is across the time frames. And the next input is memory is equal to 3. So what this input gives to the algorithm is as we said before. So in these tracks, there are certain time frames in which the track disappears and then appears again, and that can happen due to the particles getting out of focus during its movement, and this particular problem is taken care by memory is equal to 3 input here. So the when we give value of 3 here, the algorithm will even link the trajectories which are reappearing after a time frame of 3, and the algorithm will connect the trajectories between the initial and final values. So I hope this answers the question you was asked before. So I will show how the link data frame is. So this data frame has the frame values here. So for the 0th frame, in 0th frame, they have 4 particles. And so the particle heading is the identity of each particle. So 0, 1, 2, 3, and so these 4 particles are seen in the first frame. So as we can see here, on the 250th frame, the same particle which is seen in the 0th frame is present in the 250th frame. So, and its x-axis position is 57.4. So in the first frame, the second particle position was 59.2 and changed to 57.5 here. So this shows that the particles has been linked across the time frames, and their trajectories has been linked.
Badil, hi. I'm sorry. I think it would be nice if you can wrap up because we may not have enough time to go over
Yeah.
Dhruv's section. So please wrap it up in the next 3 minutes.
Yeah. So I will.
You can come to the end now. The final. I think the concepts are clear.
Yeah. So here we link the data, link the trajectories, and then the kind of trajectories which are only seen for few frames, and we remove those trajectories and include only the actual trajectories. So that's the filtered tracks here. So after the filtering, we will get the coordinates for all the trajectories, so the particle trajectories in our Kymograph. And so here in this code block, I have defined a function that will so we give the ID of the particles in our time frame data, and so this will take in the ID of the particle and plot the trajectories from the extracted coordinates using all the algorithms before. So I've just defined the function here. I will use this function in the next block. So here I will try to overlay the tracked or detected trajectories over our previous Kymograph. So this is the Kymograph we created before with the background subtraction. So here for all the particles in the particle list, this particle list is a list containing the IDs of all the particles the trajectories of the particles we detected before, and this loop will iterate to every particle and plot the particle trajectories. So here I have overlaid the extracted coordinates of particle trajectories over our actual Kymograph, and we get a good fit for the uh, tracks of the particles. And since we have the coordinates of each trajectory separately, we can use that to analyze the velocity and displacement of the particles. So the next function definition is here is to return the velocity, the maximum displacement and the average velocity of each particle tracks. So so by using that function, I have extracted the maximum displacement and the average velocity of each particle we detected before, and use that data to plot how the distribution of displacement and velocity looks like. So this is the summary. This is the things we have gone through this tutorial. So I will end here the time limit, and I will. So if there is any question, can answer now, or I can.
I think, Badir, it's a good time to take a break. Let us take 5 minutes, and in the meantime, Dhruv can share his material, and people can also ask more questions. Yes. Dhruv, I think you should take over from now. Yeah.
Hi everyone. So I think this is a good time to take a break for 10-15 minutes. So we'll be back at 15:45. And
Yeah.
then you can start, Dhruv.
Yeah, okay.
Okay. Meanwhile, if someone has some question, they can ask.
That was interesting, but it wasn't prolonged because you're still online. There's some
Welcome back, everyone. I hope everyone is there. I think you can start your screen.
Can I start or should I wait for a few more minutes?
I think you can start.
Okay. I hope everyone can see my window. This is tutorial to cell tracking dot Python notebook. Yeah.
Yes.
So like starting off with this image analysis and image processing topic, I wanted to highlight the broad field that we all are working in. So this field is known as computer vision. So this term computer vision has been popularized very recently because of deep learning techniques that have been applied to real world scenarios, tracking different types of and very complex objects. And what we are seeing in biology is that the algorithms such as Otsu's thresholding, background subtraction, and basic algorithms like that are being transferred, are being developed in this field, in computer vision field, and these are being transferred to biology fairly recently. And what we are seeing currently are the applications of CV in biology. And these are used mostly to track cells, segment out cells, quantify different cell biology data. So now these algorithms are mostly coded in different programming languages. Now, most famous of them is the OpenCV module of C++, which has like a consortium of different type of algorithms. And this OpenCV library is coded in Python. So that import module that you saw, import CV2 and import IAMUtils. So those are actually OpenCV libraries, which we are importing from C++ to Python, and they are made compatible to run with Python. And then we have MATLAB. So MATLAB is more of a service, like it's a proprietary software. So they have something called as a image processing toolbox, where you have most of these algorithms coded as functions. So it's very straightforward to do image analysis in MATLAB. And then you have Java. So Java is what runs behind ImageJ. So the J in ImageJ is Java. And yeah, so these are the coding languages that are used in the industry to develop new computer vision applications for real world scenarios. But for a biologist, most of these applications come like coded or packaged into these interactive GUIs such as ImageJ and Cell Profiler. There are many more examples of different types of tools that are available, which are like aimed towards cell tracking, cell segmentation, particle tracking, and things like that. So what I'm going to do today is take an example of a Python code for cell tracking. So it's a particle tracking algorithm. So the basics are same. The data is a bit different from cell and particle, but the basic idea is same. So I'm just going to demonstrate a Python script that does that does detection of particles. So these particles are bead. I will explain that later, and then we'll try to track them different time series. So what you saw in Kymography was that the movement of those particles was one directional. It was like from left to right or right to left. When we are talking about 2D tracking, we are referring to like a region where cells are moving up and down, and we want to like track them. So someone asked me that they are working with tracking worms. So yeah, so Kymography might not work there. So you need to be able to segment out cells and then track them. So yeah, so ImageJ and Cell Profiler, these are directed towards us biologists. So we don't want to get into complex algorithms. We only want to use them. So yeah, so ImageJ and Cell Profiler and other GUI softwares are very well like most used. So what we will try to achieve today in this Python notebook is I have this image data. So these are beads which we have acquired under DIC. So these beads are going in and out of focus. So what you're seeing in the background, in the foreground are the same object, but since they are in a different plane, their appearance looks different. And what we'll try to do, we'll try to separate or detect these black spots separately, which I have shown over here. So the detections are overlaid as circles, and then we'll try to track them over time. So first, so as for this collaboratory framework, as Dr. Chaitanya discussed, that there are like two different types of kernels here. One is just this text section where I can add text, I can separate text, I can import pictures, I can display them. And then we have this coding block where I can input different commands to download datasets. Now, since this collaboratory is an online platform, and my data is, I have shared that on a Google Drive. So I need to download the data from the Google Drive to this environment, and I do that using this gdown command. So gdown is a short for Google Down or Google Drive download. And I just pass in the sharing ID of my object. So yeah, so if I run this, it will download the image file that I'm going to use for the analysis. So it's an 80 MB file. Then there's a PNG that I'm downloading, but I won't be using that. So like I said, that most of these algorithms are coded into modules, into functions, which are packaged into modules. And since we don't want to like redo the whole coding part, what we'll do, we'll import specific modules from libraries which are shared, which are made open source, such as OpenCV, Matplotlib, and SkyImage. So these modules are used for image analysis in Python. Yeah, and I will use TrackPy to track my detected granules. Now, I think we will share this module with you later, but so if you run this section where I import all the different modules which I will use. So usually if there's a module that is missing in your installation, you will get an error. So if I just clear all output, let's run it again. Yeah, so I think TrackPy is installed in my network, so that's why it's not giving me an error. But usually when you run this code block, you will get like many errors. You will it will show that Matplotlib is missing, SKImage is missing, TrackPy, Pandas, and all these are missing. So what you have to do, you just have to install the specific module using this command, this pip install TrackPy. So if there's a module missing, you can just search on Google, like to install, followed by the name of the module, and it will just give you the command. So you can just copy paste in a module and just run it. So the first step that we'll do is just reading in and displaying images using CV2. So as we have discussed like many times, these images are made up of, these are essentially stored as matrices in the computer memory. So this is what you see in the leftmost side, and this is what the computer sees in the rightmost side. So what it sees is just a arrangement of rows and columns where each pixel has its own gray value. And you can see that there's a difference between interpretation of what you see visually and what the computer sees in its memory. And our task, like as a practitioner of image processing or computer vision, your task is to identify features from this matrix of data. So what I'll do first, since I've downloaded, so when I download the image using gdown, this video gets stored as beats 0R10031 in my collaboratory environment. So this name over here has to be passed to this function. So this function is IamReadMulti. So this is the CV2 function that reads in multi-frame TIFF files. So if you have a single TIFF file, you will usually use IamRead. But if you have a stack of TIFF files, you will have to use this function. So let's see what happens if I run this. So if I print out, if I just display what IAM is, so the way programming works is that you have a variable. The value of that variable is decided by the function that you run on the right side. So the output of this function cv2.IamReadMulti followed by the video name will be stored in this variable IAM. So if I run this again, so like I said, the image is stored as a matrix, right? So you see that there's like an array of different matrices values which are stored, and these are like different frames that are stored stacked upon each other. So this is what the data looks like of IAM. So this is like a huge data file. So I will just clear this output. Yeah, so once we read in our data, we want to check like how many frames are we dealing with and what's the size of my images. So there's like two functions here. There's this len function that just calculates how many frames I have. And there's this image array dot shape function, which calculates how many, what's my dimension of the images in X and Y direction. So it probably won't work because this variable is not defined yet. Yeah, I think I already run this. I've already run this notebook first. So I'm defining image array in the next code block. So this should ideally come before this. So what I'm doing, I'm defining another variable. I'm just passing in the values of one particular frame where n is the frame that I want to have. So before I get into this, just let me show you the output of this. So what I'm showing here is the number of frames and the dimensions of my images. So that the dimension of the image is specified as 221, 520 by 696. So 696 is my X dimension. So X dimension is interpreted as number of columns, and my Y dimension is number of rows, which is 520. As you can see, this is around 520 and this is around 696. And 212 is the count of number of frames that I have. So I have 221 different slices of this image dimension. So if I select a different frame, it will just update that particular frame. Since I have 221, I can just select 220. So it says it's out of range. That is strange. So it's IAM, it should be image array. We run this again. So what usually happens is that variables get overwritten because I'm running it multiple times, so I get into different types of errors. So let's do this again. Wow. So debugging your code is actually the most time consuming part of programming than actually writing it. So yeah, so it worked. So let me change the frame again. Yeah. So essentially when I read in IAM, I'm reading in my image object and then I'm specifying the number of frame over here. So this one is actually the place where my data is stored. So if I type in im.shape, yeah, so my im is not actually an image, it's a tuple. So a tuple has properties of length and not shape. So let me type in length. So it says that my image is of length of 2. So this usually happens because this function passes in two values. One is a Boolean, 0 or 1, and second is the variable where my data is stored. So the first Boolean just tells you whether the IAM_read function has worked well or not. So if I do IAM1 and value of 0, that should give me a value of 1. Yeah, so this gives me true. The 0th value gives me true, which means that the function has worked properly. if this image is missing from your content directory, this would have returned false, which means that you need to download it again or check the name of the image. Now, if I use the, so like as Chaitanya mentioned, that the count starts from 0. So 0 is 1 and 1 is 2 in terms of the element where it's present. So if I do im1.shape, that should give me back my image dimensions. Yeah, so it's a list, so I need to convert it to an array first and then do this. So the length of my second object in this IAM is 221, which is corresponds to the number of frames that I have. So yeah, so you can select different frames and you can show a particular frame using this IAM show function, which is a function of this module PLT. So when you are importing modules such as matplotlib, you don't want to write matplotlib every time you use it. So you can shorten them using, you can shorten matplotlib.pyplot as PLT. So whenever you are using PLT, you're actually calling this particular module. Yeah. Yeah, so since I'm running my code blocks multiple times, I'm changing the variable names quite a lot. So that's why it's giving me some errors, but it's fixed. So I'll just come to what I'm doing over here. But first, let me show you this plot. So when you see an image profile, image time series like this, you see that there are beads and which are surrounded by a background, and then you have these bright spots. So what is actually happening over here is that these beads have a value, a gray value closer to 0, and these bright spots have a gray value closer to 55. To visualize this particular aspect, the aspect of what gray value they have, I use a 3D plot where the third dimension will be coded by the gray values. So what I've done, I've taken a slice of I think 100 by 100 from this image specified over here. So I take 300 to 500, so it's a 200 by 200 slice of my image. So this is my image, this particular section, and I'm just slicing it in this dimension. So this should be rows or columns. So I'm specifying that just take a slice of from here to here. And then I'm displaying this over a mesh work. So a mesh looks something like this. Now over here, what you see is that those black spots that you saw in my input image, they correspond to these valleys in my 3D plot. And you can see the intensity value now coded as my z-axis. So it ranges from 0 to 250. It's rescaled to from 50 to 55. And what you can see is that these black spots, they correspond to these large valleys. Now, your algorithms such as thresholding, they work by slicing this 3D contour into sections of foreground and background. So what usually happens is you specify a threshold of 100. So what I'll do, I just specify a threshold of 100, and everything above 100 or below 100 is removed depending upon what region you want. Since I want the dark regions, I will remove everything from 100 or above, and I will get these dark spots. So that's how thresholding actually works. It works on the intensity gray values. But what we will do is we will do something called as image filtering, broadly known as blob detection, to detect these dark granules. Yeah. So these type of contours, they help you to get a feel for the data, like how exactly it looks, what's the spread of the intensity. So you can see that these black spots have a very narrow intensity, and they occupy a very small region of the image. So these are very small, and their spread is very less. And this type of information helps you to filter them out using convolutions, which people do. So just keep that in mind that these granules, they occupy a small area, and their spread is quite sharp. it's very sharp. It's not very broad. It drops out very fast. Yeah, so since we want to detect these dark spots, what we'll do, I'll just invert this image. I'll just convert all the values which are lower to the higher values. And this will make those dark spots as bright spots. So what you see is that I've inverted the image. Those dark spots that you saw earlier now are represented by these these bright spots. Now, so what you see that their appearance is somehow like blobs. Similar to cells, they appear as like a smooth intensity region surrounded by a background, and there are many different objects over here. Now, this data set is now to segment these granules out specifically, it's quite tricky because there are many other background things over here. There are bright spots which are broad, which have broad intensity distributions, and then there are corresponding dark spots which were earlier bright. So now we will try to segment them out using a technique known as blob detection. So in image processing, we can refer to this as image filtering, where we will filter out these granules from the background. So this is what the basis of image filtering looks like. Just 1 second. So what happens is if I take this image and in the lower row, I have just shown the similar 3D plots. So these were generated in MATLAB. These correspond to the original work that we did with Dicot, where we segmented out the granules. So this is my input image, just an ROI. And we designed a SOG kernel. So a kernel is basically something that you want to use to filter out your object of interest. So the process which is happening between image profile and your kernel is called as a convolution. I'll just explain what convolution is. So I'll come back to this later, but let me just explain what convolution is. So what I do, I will create a kernel, a 3x3 kernel with all values of 1. And then I have an image which is of dimension, what you saw, 626 and 520. So what convolution is just passing of this kernel, this 3x3 kernel over the whole image in a step-by-step fashion. And every time it makes a step, it computes this particular value at that particular pixel. So when it's at the topmost of my image, it's computing. So if my, just 1 second. So if my image dimensions are, just say 500 to 600, this kernel is passed from the top left to the bottom right of the image. And when it passes, it computes the cross correlation between that particular section of my image, and it computes the value based upon what these values are. So every time, so suppose it's at the very topmost column, topmost column of my image, it will just compute 1 into. So this is a 3x3 convolution kernel. So it is passed in a 3x3 fashion across the whole image. So I don't have an image to clearly. So if we take this example, so this kernel is first will be present over here. It will be multiplied with the pairwise value of the image and the kernel, and it will compute the sum of this kernel and this area. Okay, so if I compute my image with this kernel, which is only once, I should get an image which is similar. So yeah, so there's no change in my input image when I multiply with this kernel. So this kernel is essentially a similarity kernel. It doesn't change anything because all values are 1. So I'm multiplying all gray values by 1 and computing their average. So it doesn't do anything. If I change some of the values of this kernel and now design a kernel where the horizontal and vertical values are 0, and I convolve this kernel with my input image, you don't see much difference. So let me increase this. So what you're seeing over here is that my kernel has somehow modified my image representation. Now this kernel is defined as the mid section of both x and y zeros, and the topmost left and right regions are 1 is positive and 1 is negative. So what is happening over here is that at every place, this particular computation is being calculated. So the pixel value at the top right is being subtracted and the bottom left is being subtracted, while the pixel values at top left and bottom right are being added. So this somehow changes my image interpretation. So this results in this kind of an image, where now all the spots have convolved and transformed their representation. So what we'll do, we'll design a kernel such that the multiplication between the image profile and the kernel highlights my granule images. So to do that, what we'll do, we'll design a kernel that looks approximately like my granules. So you can see that my granules have an intensity peak. They are quite broad, and then they are surrounded by a smooth region. So what I'll do, I'll design a kernel, what we call as an SOG kernel, that approximates this type of distribution. And you can see that this is also looking very similar to my granule profile. And what is going to happen is that when I convolve this over this image, the regions where this pattern match, those regions are going to get highlighted. And as you can see from my image to my filtered image, the granules have highlighted in their position. So you can see the difference between image and filtered image. So if you do a thresholding on filtered image, you'll get an absolutely very nice results of granule. If you do thresholding on this image, you won't get any good results because there's too much background and the distinction is not clear. So what filtering does, it enhances your background from foreground. So there's a filter called as Gaussian filter. So Gaussian filter works at like blurring of objects. So if you see my video in Cisco, you can see that the background of my image is blurred. So what is essentially happening is this is the data from my webcam is feeding to the Cisco platform, and they are blurring out the background using some Gaussian kernel or something else. But usually blurring is done by a Gaussian kernel. So this is what the image filtering looks like. You design a kernel according to your object of interest, and you do a convolution, and you will see that the object will get highlighted, and then you do your thresholding. And when you do your thresholding, you then detect them by their centroids or by their weighted centroids, which will take into account the intensity distribution in that region, which is a bit more accurate. So that's how blob detection works. Now, over here I've shown, so the blob detection that we coded in our work was SOG, was this kernel that we wrote and we implemented. But in Python, since I don't want to code that again, I'm just using this package from skimage that already has 3 different types of kernels in it. So these kernels are Laplacian of Gaussian, difference of Gaussian, and determinate of Hessian. If you do if you use Image2Numbers, you will see that there's an option for filtering, and you'll see multiple filters there. You'll see Gaussian filter, and as someone mentioned in the chat, median filter and averaging filter. So I think median filter is used for denoising. It's not really used for filtering out objects. So yeah, so if I do any median filtering here, I don't think I will get any good results. So a median filter is take the median of your whole kernel in that region, right? And so if I have to code it here, so I cannot code that as a kernel, but I can pass it as a function that you take a 3x3 region and you just compute the median in that region, and you replace the value of that pixel by the median of the surrounding 3x3 pixel. And what I'll get is a median filtered image. So this is, so these are like the basics of filtering. So you design your kernel, you convolve it, and if you design it well, you will get your regions of interest highlighted, and then you do threshold, and then you do detection. Now this works mostly with blobs meaning these objects which are more or less circular. Now these type of approaches might not work very well with bacterial or cell images, but with E. coli we've had some success. So since E. coli is like cylindrical and quite symmetric, most of the segmentation of E. coli can be done by this Laplacian of Gaussian filter. You just specify a filter, you convolve it, and you'll see that the E. coli cells have been highlighted. And then you can segment them out easily. And then to optimize those segments, you use some fancy active control method. But we won't get into that. Yeah, so that's how the filtering works. So this is just an example of a random kernel. So this is, since this is a 3x3 kernel, the effect is very limited. It's only in a 3x3 region. If I designed a big kernel like a 9x9 or a 12x12 filter, I will see that the effect will change dramatically. You can try doing that. So it won't work very well because most of them are ones and only these values are different. So it won't have much difference, but yeah. Yeah, so what you saw is that at every pixel value, what I'm doing is I'm doing some transformations over here. So what I can do, I can create something called as an averaging filter. So what I'll do, I'll create a 12 by 12 filter, and I place each value as the mean of the total length. So this should work. Just let me comment out these sections. So what I now have is a 12 by 12 kernel where each value is like a divided by 12 by 12. So it's actually, it will compute the mean in the 12 by 12 region. So if I convolve this with my input image, you can see it has an effect of blurring. So it's similar to Gaussian blurring, but it's not exactly that. Gaussian blur is defined by the Gaussian distribution profile, but this is how the averaging filter works. So median filter will work on the same principles. So yeah, but we won't be using this. But you can see how filtering deforms your image. And an interesting thing is that this process of convolution is what drives most image processing tasks. Even deep learning and everything, they have these filters arranged in large numbers, and that's actually what they're doing. The difference being that since this is like a 12 by 12 filter, so this would have like 244 different parameters, and deep learning methods, they have upper of 7 to 9 million. So GoogleNet has, I think, 9 million parameters. So that's why they are very powerful, because they can transform your input image into different interpretations and then choose out which interpretation works well for classification task. But we won't get into that's it. Let me just hide this. So how do you decide the blob diameter? So once you have an estimate of what your granule diameter is, you can decide your kernel profile according to that. Okay, so what I have observed is that even if you miss by 10, 20 pixels, you will get good results. But a rule of thumb is this diameter should be equal to your object of interest. And again, that causes problems, because if your granules are of different sizes, you might get into trouble. But I'll demonstrate that in later sections. I think I should speed up. Yeah, so here what I'm doing, I will specify blob detection kernel, difference of Gaussian specifically. So difference of Gaussian is this kernel over here. So this image is actually a space image where they're detecting galaxies and they're counting number of galaxies and stars. So anyway, so I will define a kernel, and like I showed previously, that a kernel is defined by two different parameters. One is the sigma value and one is the filter value. So blobdog for some reason doesn't allows me to specify the filter value. It should somehow, I think, in the documentation, but I haven't done that over here because I didn't need it. And it specifies a threshold. So threshold is this step, the filtered image to the segmented image. So I've set these values to 1.5 and threshold of 0.05. So let me see what happens when I do convolution over here. So what I'm doing, I'm overlaying my detections over my input image. And what you can see is that I don't really get anything. These detections are all over the place, and I'm not locating these dark granules. So let's see what we can do to fix it. So my current image. Let me check my variables are defined correctly or not. I think they are. So let's first just reduce the threshold or increase the threshold. So now, as what you can see over here is that specifying a set of parameters of sigma of 2.5. So sigma of 2.5 is still low. And threshold. So I will just show you how changing sigma changes my output. So right now what you can see is that these dark spots are being detected very well. You can see that I'm not getting confused by these dark spots, which are very broad. So if I want to detect these and not them, I will specify a higher threshold. So if I increase my standard deviation of my filter, should be getting detections around those dark granules too. Yeah, so as you can see, now it's been thrown away by background a lot. So when you are playing with image filtering and convolution in general, what you need to do is you need to just optimize these two values. You need to optimize your sigma value and you need to optimize your threshold. What Dr. Chaitanya showed in the morning is that he showed some F1 score metrics, some ground truth metrics. So what we did, we annotated several granules in different ROIs and we iterated over a range of values. So thing with Python is that instead of specifying values one by one, I can just write a range of values that it can iterate through. I can write for all purposes like 10,000 different values of this, 10,000 different values of this. I think 10,000 is the overstatement. Maybe 100 or 200 values. And I can just pass on these values and just compute my detections one by one. And what I can do, I can automatically compare my detections from the ground truth and I can compute which set of parameters give me maximum F1 metric. So that's how filter optimization done in the previous lecture. So anyway, so now if I have these parameters 2.1 and 0.1, I see that my detections are more or less very good. I'm getting these small dark spots and the function that I used was this. So this is actually convolution only, blob dog, and this is my image, this is my sigma, and this is my threshold. And these set of codes are only for display purposes. I'm not doing any computation over there. This is just to display these circles, display the image, and I'm just iterating over all the objects that I found. So what I'll do, I'll just pass these set of parameters throughout the whole time series. So as we saw that there were 221 different frames. So what I'm going to do, I'm going to pass these parameters. I'm going to pass the same function over the whole time series. So it might take a while to run because there are like 221 different frames. I think I have 10-20 minutes more. Yeah, so I think this will take a while. I can answer the questions then. Okay, so it has finished running. So I was storing all the data points in this frame called as object data frame. So if I run this, you should see a set of X and Y coordinates, their radius and their frame number. So since I said the frame number starts at 0, you'll see that the count starts from 0 and ends at 220. And what I'm seeing is that these set of coordinates. So at every frame, I have a set of detections. I have a set of coordinates in X and Y, which correspond exactly to the center of this circle. So these are my detections. So what I'm going to do again, I'm going to link these detections throughout the frames. And I did what Badhi did earlier. I'm going to use TrackPy package for linking. You can code your own tracking algorithm, but that is, I think, outside the scope. We have done that in MATLAB, but in Python, I don't think we need to do that. So I'm going to call this very powerful function tp.link. So tp is my module again, the TrackPy module. And I'm going to link all the detections in this data frame. And the detections, like I said, are based upon a nearest neighbor. So if we see this schematic, the objects in red show the detections in the current frame or the frame number 1, and the blue are the detections in the frame number 2. And so these tracks are linked based upon a nearest distance metric. And there are like two parameters over here, which are 18 and 5. So 18 is my gating threshold. What that means is that if the nearest detection is outside this 18 pixel value, it won't propagate that trajectory anymore. And what memory means is that if my trajectory is less than 5 frames long, it will reject it from my overall analysis. So these type of metrics you can play around depending upon how fast your object moves. And you can visualize the results to see if those metrics work well or not. So I'll just run this tp.link. It runs through all the frames, 220 frames, and it says that there are 48 different trajectories are present. Can we measure the localization precision of any particular molecule using this module? So localization precision will depend upon a ground truth. You need to specify what exact location of that object is, and then you can compare it using a distance metric, like a simple distance metric between your detections and your ground truth. you can opt. So the detection is optimized usually by some kind of a Gaussian fitting. So once I get my detections, I can fit a Gaussian to this region, and I can specify the exact peak of this region using Gaussian fitting. Exact peak that corresponds to the deepest point. Yeah, but it's not very straightforward. For this, it's easy because the profiles are very Gaussian-like, but for some objects, it might be difficult. But yeah, for increasing precision or localization or to achieve sub-pixel accuracy, you need to do some kind of a Gaussian post-fitting to get an accurate estimate of the position. Yeah, so once T is done, I'll just plot what the trajectories look like in the figure. So this is what the outcome of track linking is. Now, since collaboratory is not very suitable, suited for opening images, I mean, not opening images, opening displaying time series, I'll just show you what the outcome looked like when I used it in my Python. So this is the tracking outcome from this script only, not the script. This is a similar script coded in MATLAB, but all the basics, all the process, all the values are all the same. So what you can see is that now I can track these bright spot very well. And once I have the trajectories, I can compute their displacement, their speeds, their mean square displacement. I can get an estimate of viscosity. I can get an estimate of if the things are drifting or not. I can get an estimate if like how fast things are moving and whether the motion is ballistic or not using post analysis. So I'm not going into the post analysis stuff. I'm just showing what the tracking output looks like. So you can see that we have very good tracking results. So we used all these similar principles for our C. elegans movie. So we designed a pipeline in MATLAB. So this was not coded in Python. We designed it in MATLAB and to segment out these lipid granules in the C. elegans embryo, and we could track these granules over time. I'll just show you the movie first before going to the details. So this is a C. elegans embryo, which has been tracked using the software. Similar to what Sir showed earlier. Can you guys see it, right?
It's loading, Dhruv. It's somehow still loading. It's showing the spinning.
Yeah, it's still loading on my screen too. I think this is the large one, maybe. No, it's this works.
Yes, it's now showing. It was until seconds ago.
Well, it's a very nice movie, so I do want to show it.
Dhruv, while it loads, maybe you can take the question from Shomin, which is, can we
Okay.
measure the localization precision for any particular molecule using this module like NENA? What does that mean? I don't know.
So for the case of localization precision, like I said, that it has to be compared with the ground truth. You need like an expert or like a very good data set where the position is very accurate. Nearest neighbor analysis. Okay. Measure the localization precision. So I'm confusing localization precision with detection precision. I think you mean something else. So can you please go into the details? Because for me, localization precision means that how accurate my detection and the actual position of the object is. And the linking is done with the nearest neighbor analysis. So I don't know. So if you're asking how accurate the nearest neighbor linking is, well, it's the most simplest one and it's prone to many errors. But so the tracking field itself is a very big field. There are lots of algorithms that can take into account jumps, skips, and which prevents spurious linking. But yeah, so Somen, if you can elaborate on your question, maybe I can add more details. Well, this is still loading, so I'm just going to download this. That worked.
You can unmute and
Yeah, it's
ask the
working.
question.
Yeah, so I'm going to stop and share this video. Just 1 second.
Yeah, I think
Can
as someone just said earlier, if you have questions, this is a good time to either raise your hand or even unmute. Yeah.
So can you see the screen?
Yes, now I see it.
So you can see that these are granules. I have separated these granules in anterior, posterior, and middle for some distinguish between different regions, and then we can see that we can track these granules using the same nearest neighbor algorithm and Gaussian filtering. And from this, we can estimate what the intracellular dynamics look like, because because we have not done any labeling, we have not done anything. This is purely from DIC data. So this is like the most accurate description of intracellular dynamics that you can get. So that's how we transferred these computer vision applications to our purpose. Let's share that notebook back. Yeah, so yeah, the basics were same. We did filtering, we did detection, we optimized the detection based upon the granular intensity. So we use something called as regional maxima. So after we get our filter object, we compute where the intensity peak is. So that improves our accuracy of detection, and then we track them using this similar gating method. Yeah, so summary of this talk would be like in Python, how you can read and display images, how you can apply different types of blob detection and filtering modules, and how you can track these detections in TrackPy. So if you have any questions, you can like ask. I think that should
Can I
be
ask?
Yeah.
Hello, Dhruv, can you hear me?
Yeah.
So yeah, actually my question is not related to this one also.
Could you just please tell us who you are, because we can't make out otherwise? Yeah, just identify yourself while you ask, please.
This is Soman. I am actually from Sarid Agastya from JNCASR.
Very nice.
So actually, like since people are presenting about this mandem, actually I am more interested to, because I am now doing some analysis, so I am now facing some problem about that one. So the problem is like people have done like actually mainly I am working on some supernuisance microscope. Okay. So single molecule tracking and also some paint-based microscopes.
Yeah.
They are people have done the localization creation using this nearest neighbor analysis. Okay. So nearest neighbor, whatever like Dhruv showed, like nearest neighbor, they are using nearest neighbor algorithm to uh, uh, distance between two nearby molecules.
Yeah.
Okay.
Yeah. Yeah.
So that's why just I asked whether from using this algorithm we can do this localization pressure measurement using this nearest neighbor.
Yes. So what you have is like between frames, you have like a set of detections, right? So if like there are like 10 particles in frame 1 and 2.
Yes.
So what you
yes.
do is you compute the distance between all the pairs, between
Yes.
everything, between particle 1 with all the other 10 particles, and you optimize that distance matrix. So you can do that optimization in MATLAB. I'm not sure if Python should have, absolutely, but I've done it in Python. So what you do is you optimize that matrix, you link them such that the global minimum distance is the least. You can either do it in a global fashion or in the very local fashion, where locally you just link one granule to the nearest granule in the next frame. So that's how nearest neighbor detection, not nearest neighbor, it's just nearest detection. Nearest neighbor is something related to machine learning. So I'm not really sure what that paper is about. So there might be some things that are different over here, but this is just a nearest detection method. Nearest neighbor, maybe, yeah, we can say it, but yeah. Yeah, but it's very basic to particle linking. This is the most basic algorithm that you can code. After this, there are multiple different like Kalman filtering, which go into prediction of where the particle will be to optimize the linking. You have Hungarian algorithm and many different algorithms. So this is the most basic one. And this works mostly because it's very efficient. Since we had a lot of data, we didn't want to like, optimize our tracking and everything. So for the initial starting point, since we have like more than 1,000 granules in one particular frame, we just link them based upon our nearest neighbor criteria. And since our detection accuracy was quite high, about 0.9, we didn't have any spurious linking problems as such. As you saw in the video, also the movements were quite smooth. Yeah. Any other question? Yeah, I don't think we have another. I think I have not introduced myself. So myself, Dhruv. I've worked with Dr. Chaitanya, third year PhD, and yeah, I work with image analysis and all this stuff.
Everyone, if we have no more questions,
Your sound is breaking up. I'm sorry. Could you
then
just repeat
I
that, please?
think we'll so I was saying that if there are no more questions,
Yes. Yes. Correct. Mhm.
can everyone hear me now? Yeah. I was thinking that if there are no questions, then we can end the session. So
Yeah.
I would like to thank Dr. for teaching us how to write basic code using Python and showing the programming for kymography and cell tracking and analysis. And I appreciate your patience watching the queries and clearing of the pattern. So for participants, we will be sharing the link for today's recording so that you can practice writing your own computer programs to analyze your data. And thank you participants for active participation and making this event successful. So now I will request you all to switch on your cameras. I'll take a screenshot of it.
Dhruv, I think maybe, yeah, you
Yeah.
can stop sharing.
Yeah.
Should we smile?
Okay, everyone. Bye.
Oh, I see some familiar faces here suddenly. Hi, Kiran. Okay.
Is it over?
Yeah. So I'm going to take your leave. Thank you very much, all those who attended, all those who organized, and I look forward to any feedback if you have any. We will keep the Google Classroom. Will have some more material uploaded after this. So Dhruv, I, and Badira are going to talk it over. And thank you for attending, and best of luck for the rest of the course, which goes on, I think. Okay. So see you then.
Thanks,
Bye.
Chaitanya.
Bye. Bye. Hi, Lakshmi. See you later.
Bye. Yeah.
Thank you so much.
Thank you.